So it's a pleasure to introduce Nick Sauter from the Lawrence Berkeley Lab, who's going to be telling us about DIALS.
Thanks. And thanks to the organizers for the invitation. Can you all hear me? I'm going to be talking about DIALS this afternoon. DIALS is hopefully the future of data reduction. It's a fairly large collaborative project between my group at Berkeley Labs and folks at Diamond and CCB4, and lately Herbert Bernstein at Baldwin College. I'll talk about that in a bit. So what is the future of data reduction? Well, certain DIALS stands for Diffraction Integration for Advanced Light Sources. And certainly, as we've heard about, we want to be optimized for rapid data collection on pixel array detectors, as we've heard. And we also want an extensible design for new algorithm development. And as I'll talk about a little bit later, there is a relationship between DIALS, which we think of as being primarily targeted at synchrotrons, and also the efforts in my group to process XFEL data as well. So when we started this a few years ago, what was on the horizon was the Pilatus 6M, and we were looking at framing rates of 25 hertz and half a terabyte per hour of data collection. And of course now, with the Eiger on the horizon, we're now talking about 10 terabytes per hour, and other detectors from ADSC also contending. So I think it's clear from this that the future is that we're no longer going to be processing this amount of data manually by interacting with a graphical interface. That, in fact, what we expect is at the synchrotrons, data sets will be processed, at least for the first pass, by some automated process that's running on a server. So that's what DIALS is geared at. Also, we've really seen an evolution in the way samples are delivered. Now we have in situ data collection where we can put the entire sample, the entire chamber where the crystals were grown into the X-ray beam. Also the ability to scoop up slurries of microcrystals onto a grid and stick them in the beam. And what I'll talk about later with XFEL is we're delivering samples by a liquid jet. So clearly there's no expectation anymore that we're going to have a single crystal in the beam. We have to contend with multiple lattices that we observe in the diffraction pattern. I liked the slide from Phil yesterday where he talked about the merging process, where we don't measure the structure factor directly, we infer the structure factor from measurements of the intensity. I guess what I would say for this talk is that in data integration, we don't measure the intensity directly. We infer the intensity by measuring the pixels on the detector. And it's a very complicated process. We have pixel measurements, and then we have a very complicated model, and I'll talk about all these things in a bit, from which we infer the intensity. So everything in my talk today is geared toward the model. We have to develop the best model that we can to get the best intensities given the data that's on the pixels. So from that, hopefully we can, as I say, model and get good intensities from normal Bragg spots. But in the future, there's other things too. There are other lattice phenomena. Here you can see these Bragg spots have satellite peaks. This is all published stuff. This results from incommensurate modulation. This is where in the crystal there's sort of a displacement wave that's going through the crystal, but the period of the wave is not commensurate with the unit cell dimension. That gives satellite peaks. These are all published cases. This is lattice translocation disorder, where successive layers stacked in the crystal have a small translation in a random dimension. So we would like to be able to use physics-based models again to get the best possible Bragg intensities from these types of diffraction patterns. Clearly, this is still on the horizon, but what we want to do when we're developing DIALS is not to lock ourselves in by making simplifying assumptions that would preclude putting in this new physics later. So we want to be able to put in, we want to be able to have an architecture where we can slip in new types of models in the future. And thinking about stretch goals, this is diffraction from photosystem 2 that I'll talk about later. Look at the Bragg spots. They go out to about 4 angstroms here. But there's signal. It's thermal diffuse scattering that goes out way past the Bragg spots. And in the future, we'd like to understand this and to understand that the diffuse scatter tells us something about correlated motions between unit cells and even correlated motions of atoms within the unit cell. This is really all future stuff, but we'd like to be able to be in a position to analyze it. So this is a little bit about the software architecture of what we hope to do. It's going to be based on the CCTBX toolkit, which has been around for over 10 years. It's at the core of things like Phoenix and Label It, and it provides a good interface that lets us deal with structure factors, space group symmetry, and unit cells. And in preparation for DIALS, what we've added to CCTBX is something we call the DXTBX, the Diffraction Experiment Toolbox. And this lets us model as software objects concepts like the detector, the source, the crystal, the goniometer, and the scan. So this is our data structures that we're going to use. And on top of that, DIALS, which is actually in a different software repository, right now encodes the different algorithms that we're going to be using to process data. So let's focus in on these data structures for a moment. This sort of shows the general workflow. So we start with raw images from the synchrotron, we import them, and in the end we get integrated structure factors out that we can put into a program such as AIMLess for scaling and merging. The first step, the actual reading of the images, is obviously it's going to be different for different detector types and potentially even for different beamlines. So say I have a beamline that's special because I have a reverse phi rotation or the detector is upside down. The way we've written this is that DXTBX has a class called the format class, which understands information that's in the header in the images and creates a representation of the experiment in terms of beam, goniometer, detector, and crystal. So the experiment is fully described inside the program it's a full vector representation of everything in the experiment. And if you are a beamline developer with a reverse phi, you can simply develop a specialized format class that may have a few lines of extra code that says that this rotation is upside down. So it's written to begin with the idea that users or beamline scientists will modify it. And just to sort of pursue that point a little bit further, these are the normal processes we usually think about when integrating data. We index the diffraction pattern to get a model, and we refine the model against the data, and then we integrate the data, and then we export it. So the way it is now in DIALS, there are 3 different algorithms for indexing and 4 different algorithms for integration. So it's already a modular architecture, and it could be further modified in the future. So let me talk a little bit more about what's in these models. As I said, to predict where, so again, we start with the data, we develop a model, and then we refine the model. What does the model tell us? It predicts where the Bragg spots are, and at a level of detail where we can tell which pixels to look at and which pixels are assigned to different Bragg spots. So the parameters in the model have to do with the beam, so the S0 vector, which is the direction of the beam, and also the wavelength. So there's at least 3 parameters there. There's the crystal, so that's the unit cell, and 3 degrees of freedom for the rotation of the crystal along with the mosaicity. And the detector, well, that's pretty complicated. Here, what we're showing is the detector is a block of sensors, but it has a translation from the crystal, so that's 3 degrees of freedoms, and also 3 orientations. So that's our representation of the detector. And just to sort of drive home that point, that we really have 3 rotational degrees of freedom for the detector. So if the detector is slightly tilted in our image viewer in CCTVX, we actually represent that. So can you see this pixel is slightly tilted? And that's the way we draw it on the screen. Yeah, so we don't think about the detector as sort of this monolithic block. It's something that can be rotated in space. Also, as I keep saying, we can adapt any of these models to encode specific circumstances. So this is a special detector built by Vectris for beamline I-23 at Diamond. This is a sulfur phasing beamline, and because we're using those long wavelengths, the crystal has to be put in the center of the detector because the diffracted rays go off at all angles, and it also has to be done in vacuum. So the crystal sits here, and there are 120 silicon sensors arrayed in a cylinder. And in this case, we don't simply refine the 6 degrees of freedom for the detector. We independently refine 6 degrees of freedom for each of those 120 panels on the detector. So there's a hierarchical relationship between the detector and then the panels on the detector. This modularity extends throughout DIALS, and here I'm going to show a little bit about the way we develop and refine the model. So again, the model, as I said, consists of a beam, detector, and crystal representations. And then in order to refine this model, we have a target function. And the target function measures what the model, where the model predicts spots will be versus where we actually observe the spots. And we take this, we develop a target function that will minimize the square difference between those two things. So delta x squared, delta y squared, and delta phi squared. And we adjust the parameters in the model to minimize this target function. Now, what I've just described is for a normal rotation data set that you get at a synchrotron, where you rotate the crystal along the phi axis. Well, what about for X-bell work, where we take still shots? Well, there, the target function is a little bit different. So we have an ability in DIALS to have either a target function for scans or a target function for stills. And then the minimizer that we use is designed to be appropriate for the situation. So we often like to use nonlinear least squares minimizers, such as Levenberg-Marquette or Gauss-Newton. But in certain situations, when the matrices become very large and there are thousands of parameters to be refined, that runs up against memory limits. So there we can use limited memory algorithms. So again, just driving home the modularity of the system. So now I'm going to show a little bit about what results from model refinement. Now, some integration programs that I've had experience with over the years, it seems like you start with image number 1, you develop a model, and then you start integrating the data. So every few frames, the program goes back and re-refines the model because there could be radiation damage and the crystal can slip and so forth. So it works in a sort of a chronological workflow. But we're not doing that with DIALS, and there's a few reasons for what we're doing is we're refining them all at once, all the data in the rotation scan, we refine at once. So some of the reasons for doing that is that doing it this way recovers from the poor determination of some parameters. For example, if you have a very large c-axis, and the c-axis is into the beam, it's a little bit hard to measure that unit cell dimension. But if you rotate 90 degrees, as you would do normally in your scan, you can get a good measure of C. It reduces some of the parameter correlations, such as, is it a crystal rotation or is it the rotation of the detector? The detector isn't completely square. So those parameters are correlated for certain crystal orientations, but not for others. Certainly, it allows us to increase the number of observations that go into the refinement of any given parameter, giving us more accurate values. And also, it sort of goes along with our intention to be able to handle a lot of data at once. So we want to do things in parallel. We want to have a processor that has, for example, 64 CPU cores, or even a cluster of processors where we have hundreds of cores available. And in a situation like that, some of the algorithms in data integration can be easily parallelizable, such as spot finding. So you can easily put each image onto a separate core and very quickly get all the spots in order to use for indexing. And also the integration step, we can easily parcel out different images to the integrator. The model refinement, I think, has to be done all at once. So that's sort of, we do spot finding in parallel, we then refine the model centrally, and then we do integration in parallel. At least that's the way it is now. Later on when we want to really think about diffuse scatter and something that's really, some models that are really way out there, I think it'll be good to have this kind of architecture. There we could use possibly graphics processing units to accelerate the process of modeling. So those are the arguments for doing global refinement of parameters. But now you're going to tell me, well, okay, this doesn't make sense because the static model of the crystal is no good. And here's a good case in point. This is a thaumatin data set collected at a diamond light source where we've intentionally irradiated it with too much radiation. The resolution goes down over time. This is 720 degrees of data collection at full transmission of the beam. So the resolution starts out very good. It gets poor. The number of spots in each image decreases over time. So what we do in a situation like this is we refine a scan varying model. So the model itself changes over as a function of the scan of the image number. And right here we're changing the orientation and unit cell parameters as a function of time. And then we smooth out the results to avoid discontinuities. So some results from this, we tried it both ways, with a static model that just has one set of parameters for the whole data set, or a scan varying model. In the static case, the target function, this is the root mean square deviation in x, y, and phi, I think in units of pixels and images. So this model is fairly poor, just a static model. Then the scan varying refinement, it improves things. So we did this first and then this. So this shows that we can actually get a better model by refining a time-dependent orientation in unit cell. And this just shows the behavior of those functions. So these are the three independent crystal rotation parameters. These are sort of periodic. They have a periodicity of 360 degrees, which I guess is expected. And then the unit cell parameters, the A and the B remain fairly constant, but the C-axis increases over time, as you would expect through radiation damage. You're evolving hydrogen gas and the crystal is expanding. So that's good. Now I'm going to say a little bit about the algorithms that are used, just to briefly touch upon them. For spot finding, it's a very simple idea. We start with the raw data here, then in tiny little windows, just a few pixels in size, we calculate the mean, the variance, the mean over the variance, which is we call the dispersion, and then we take this dispersion map and we threshold it to find spots that we're going to use, and then take the centroids from that. And there are variable parameters in this. You could say the thresholding level is a parameter that the user could set. So I would imagine that there's a lot of little parameters to fine-tune things. This may not be a completely automated process, but we expose parameters like that to the user at the command line level. So you can either write it in the command line level. Right now you could say either dials.process, which is just a single command, or dials.process threshold level equals blah, blah, blah. And that's how it'll work. The integration, we actually just took what we know to be successful from MOSFilm and XDS. In the future, there may be other algorithms, but we have to start at present from what we know and make sure that works. So for two-dimensional profile fitting, that is fitting each image separately and then later merging consecutive frames to build up the Bragg spot, we use the Moss film algorithm. So here, the crystal mosaic spread and the beam divergence are used to estimate those pixels that belong to the Bragg peak. Then we look at different Bragg spots in the same area of the detector, and we take background subtracted reflections, we interpolate these, and create a reference profile centered on the predicted centroid of the spot. And then for integration, we take a weighted average of nearby reference profiles and perform a least squares fit of the profile to the actual data. And then we get a scaling factor, and that's the measurement of the intensity. Also, for three-dimensional data integration, we use the algorithm that I described to us yesterday. For this, we have a three-dimensional box, so an X, Y, and Phi, and it's transformed in a way, how did Kai say it? Each box containing a Bragg reflection receives a different transformation such that the profile shapes are very nearly identical from Bragg spot to Bragg spot. So this is a series of 9 images, and the profile, this is the center image with most of the reflection intensity, and these are the profiles for the side images. And, the background subtracted reflection is mapped to reciprocal space, and then we average and fit these profiles. So I'm going to show a series of slides that in some way shows the accuracy that we're able to achieve using these methods. So this is what's inside of a Pilatus detector. It's a 5 by 12 array of silicon sensors, as we heard this morning. We normally think of these modules as being arrayed on a perfect grid with integer pixel spacings. So between the rows, there's 17 blank pixels, and between the columns, there's 7 pixels. But this isn't really the case. There's small imperfections in the array, and these imperfections, they're calibrated at the factory. And when the detector sells the equipment, they actually provide a correction table, but the corrections are small. They're less than a pixel. And so, correct me if I'm wrong, I don't think that the normal data processing programs take this into account.
No. We could take if this information is provided.
Well, it is.
Information is simply not provided as input.
No. This is provided by...
Okay, there is a format to put it into Dental.
Yeah, right. And I think...
The information has to get there from somewhere.
Yeah. All right.
Someone doesn't input this information.
Well, maybe what I'll show provides some push or stimulus to actually to get this. And I don't know what the situation for XDS is. I think it can...
there also.
Yeah, it's there also. So this is what Dextrous provides. So this is, for each module, it shows what is the X translation that you need to bring the module in this perfect alignment, and there's a similar file that shows the Y translations. But as I said, we typically don't use, so what I'm going to show first is this is the result of DIALS integration when these corrections are not applied. And I have to explain what these color codes are. The saturated red color indicates that the correlation between the model three-dimensional profile and the actual data is a high correlation. So this is exactly what you would expect. With the bright spots near the center of the detector, the model fits the data really well. And at the edge, where there's fairly weak data or lower intensities, the correlation isn't that strong. So that's what you would expect. But a thing to draw your attention to is that you can actually see the boundaries of the modules in this data. So this is slightly less correlated than the panel that's immediately adjacent to it. So it means that these small translations really matter. So that's, integrated intensities. The other thing we can look at is simply the X-dimension residual in that target function I was talking about. So it's the agreement between the predicted spot and the observed spot in terms of position. And sure enough, it's the same thing. You can see that different modules have different residuals. And in fact, if we compare now the information supplied by the manufacturer, it's an exact match. The color coding is different, but this is the residual fit with DIALS, and this is what Dectris has already told us about the X translation. So it sure enough is a fit. And then we can use these corrections that we determined from modeling and correct it. We can parameterize the detector now. Instead of thinking of the detector as a monolithic block, we've divided it into 60 independent panels, each with full three-dimensional translation and rotation. And once we model the data like that, the residual becomes 10 microns, which is a small fraction of 1 pixel. So it's pretty encouraging. I think this is what I want to see for data integration. So nearly at the end here. So some quick results. This hasn't been used by anybody else. It's just been used by the developers. But my colleagues at Diamond collected a few data sets. This is glucose isomerase. It's integrated with DIALS, 3D profiles. Then the map was calculated and refined with CCB4 and RefMac5. So this is a difference density for the, I believe it's the ligand. This is a DNA ligand complex phased by ruthenium SAD, solved by ShellX, and merging statistics from AIMLess. And I think what we're seeing here is, so these two panels of results pertain to the DNA ligand complex, and this is output from DIALS.analyze output. So this plot is just the same type of plot that I showed before. It's the reflection correlation, it's the correlations between the profile and the actual data, showing that the strong reflections in the middle are highly correlated. And this is another way of plotting it. The sigmoidal shape just tells us that at high intensities, there's a higher correlation with the model. And I think that's exactly what you would expect. So that's where we are on DIALS at present. When can I use it? Well, it's an open source project. It's there for anybody to see, and in fact, anyone can contribute to it, or you could take it and modify it for your own purposes. So it is open source, but beware, don't go out and download it right now. It's still under development, and it's not really intended yet for general use. But we expect to have a web page sometime this year where we actually prepare real downloadable binary bundles. People can download it then, and it will be for what I would consider alpha users. So we expect people to use it and then try out some data sets and let us know what happens and what could be improved. There is already a manual. It's in a PDF format. You can download it now, but it pretty much just helps you through the process of running DIALS on one of these Simonton data sets. But I think by the time we have a web presence and we're ready for people to download it, there will be a wiki. So the wiki will be both for developers to write down, how to solve this problem, and also for users to document what they found, what are the tips that people discovered. So I think the only thing left to do is to acknowledge a whole bunch of collaborators, both not only the postdocs in my laboratory, but also sort of very long-standing support for CCTBX from Paul Adams' group and people at the Beamlines, Peter Zwart and James Holton. Herbert Bernstein's done a lot of work with us about working with file formats and getting them into a state, which I'll talk about actually later today, where we can represent in the file header all the information that we needed in the future to reconstruct the experiment. And of course, my collaborators at Diamond Light Source, who wrote a lot of the code, as well as David Waterman there, and also folks at the MRC have given a lot of feedback on the project over the last couple of years, and also Jim Pflugrath. The thing I didn't mention about this before is that it's really two groups working together with completely separate funding on similar goals. So my group is funded by the NIH. It's actually a Protein Structure Initiative R01 grant. And the folks at Diamond and CCB4 are funded by the European Union. But we work together sort of on a constant basis. So I'll take any questions.